Dear students, in today's lecture, let us make an attempt to know about insect vectors. A vector is an organism that carries disease causing pathogen or parasites, transmits these organisms and cause infectious diseases among living beings. Few vector species are hematophagous, which feed on blood of their specific animal hosts during different stages of their lives. However, many vectors are herbivores, which transmit pathogens or parasites to various plant species during they feed on plants. Thus, vectors introduce a pathogen such as a bacterium or virus or fungi or parasites into a plant or animal to cause an infection. Much research on vector transmission seeks to understand the transmission process so as to explain why only certain kinds of insects can serve as vectors and to identify what factors are required for transmission, because insecticides applied to kill vectors frequently fail to control the diseases caused by the vector borne pathogens. A good understanding of the relationships between vectors and the pathogens that they transmit is important. Phylum Arthropoda forms a major group insect vectors which cause diseases in animals and plants. The insects which become vectors to many animals including humans are mosquitoes, flies, lice, fleas, louses, assassin bugs, etc. transmits a different type of infectious diseases among humans and domestic animals. Similarly, many insects become vectors to plants and they include aphids, thrips, beetles, grasshoppers, leafhoppers, airwick, wasps, which transmit viral, bacterial, fungal and mycoplasma diseases in various wild and cultivable plants. Any insect species, if it wants to act as vector, it should have following essentials to spread infections among animals including human being and plants. They are proper pathogens or parasites delivery system, example modified mouthparts, directly to the bloodstream in animals or vascular system or cell sap in plants, animal to animal or host to host movement to provide suitable system for pathogen or parasites to survive, seeking suitable entry points for the introduction of pathogens or parasites into the body of host, namely wounds, burns and catheters, providing suitable site, example salivary gland or gut for the proliferation of pathogens, multiplication of parasites sometime when they are in the vector's body, evolution of successful relationship with the pathogen or parasite. Now, let us know briefly about plant insect vectors. The insects which belong to the order Hemiptera, Thysanoptera, Dermaptera, Orthoptera and Coleoptera are known to have innumerable number of vectors which cause various diseases in plants by transmitting bacteria, virus, fungi and nematodes also. Most vectors of plant viruses are sucking insects which are present in the order Homoptera. Their small stylets allow vectors to introduce virus into plant cells or vascular tissue with minimum damage. Example, aphids, they transmit greatest variety of plant viruses. Order Hemiptera includes aphids. There are more than 30 species of aphids act as vectors, carry wide variety of viruses. These viruses cause different diseases among the plants at different parts of the world. Order Thysanoptera includes thrips. Only a few viruses are transmitted by thrips, but their economic impact is internationally important. Franklinella species are carrying tomato spotted wilt virus. These viruses cause spotted wilt disease in tomato, tobacco, dahlia and pineapple in Africa, Asia, Australia, Europe, North and South America. Order Coleoptera includes flea beetles, mustard beetles, corn flea beetles, cucumber beetles. Order Dermaptera includes airwigs. Airwigs such as Forcicula auricularia carry turnip yellow mosaic virus and cause turnip yellow mosaic viral disease in turnip, cauliflowers and cabbage at different parts of the world. Along with all these insect vectors, certain species of wasps, bees and ants also act as vectors known to carry bacteria to cause fire blight disease in around 90 species of orchard trees and ornamental plant species in North America and Europe and other parts of the world. The insects belong to the order Anoplura, Hemiptera, Diptera and Saphenoptera are known to have innumerable number of vectors which causes various diseases in humans and domestic animals by transmitting bacteria, virus, fungi and nematodes also. Order Hemiptera. It includes bugs. Few species of the subfamily Redividae includes Quenonous bugs, kissing bugs, assassin bugs. Most of these species are hematophagous, feed on vertebrate blood and a very few species feed on other invertebrates also. Order Anoplura. It includes louse. There are 3 species of louses which live as parasites on human body at different parts of the world. Of all, only body louse is important and act as vector for various diseases. Order Diptera. It includes flies. There are many fly species known to act as vectors for various dreadful diseases around the world. They are mosquitoes, horseflies, deer flies, tease flies and sand flies. Now it is time to know about the transmission of pathogens or parasites. During vector feed the host animal blood, the pathogens enter the blood stream of the host. This would occur in many ways. Vector insert its delicate mouth part under the skin and feeds on its host blood. The parasites or pathogens are usually located in the salivary glands. Salivary secretions are used by mosquitoes to anesthetize the host. Therefore, the parasites are transmitted directly into the host's blood stream. Few species, example sandfly and blackfly, chew the host's skin and form a small pool of blood from which they feed. During this act, they transmit pathogens into their host's body. Generally, vector acquisition of pathogen or parasites increases with the time spent feeding on infected plant sources of the pathogen or parasite. There are 3 ways by which transmission would occur. They are non-persistent transmission, persistent transmission and semi-persistent transmission. Non-persistent transmission. If the rate of transmission drops to near 0 within a short time in terms of hours, in this case pathogen generally decreases rather than increases with sustained feeding on infected plants. Semi-persistent transmission. It occurs for a few days after acquisition of the pathogen. Non-persistently or semi-persistently transmitted viruses are no longer transmitted once the vector has molted its exoskeleton. Persistent transmission. In this case, the vector can transmit pathogens over many days or few weeks or even months together. For example, the beet curly top virus BCTV has only one vector. The beet leafhopper Cruciferae tenellus infectious beet leafhoppers transmit the virus after a latent period of only few hours and continues to transmit for days to weeks. The length of time they continue to transmit depends on how long they feed on a virus infected plant to acquire this Gemini virus. Beet curly top virus is an example of a persistently transmitted virus that is circulative but does not multiply within the vector. This virus also requires a longer feeding period for vector transmission. Most lengthy latent periods result from the requirement of the pathogens to multiply within the vector to attain the numbers needed to complete the vector's transmission process by reaching the salivary glands in sufficient numbers. Reoviruses, example rice dwarf disease, multiply within both their insect vectors and plant hosts. Multiplication and circulation of the pathogens within the body cavity, that is hemocoel of the vector, ensures that the pathogen will remain within the vector after molds that occur as the vector develops. Vector transmission processes are usually complex, where simple mechanical transfer of plant viruses to plants on the tips of vectors, stylet like mouthparts, takes place during feeding. Many plant parasites are transmitted by insect vectors after multiplication and circulation throughout the body of the vector. It is based on the transmission opportunity or transmission efficiency or vector competence. Transmission efficiency can increase or decrease over time after a vector first acquire a pathogen, usually by feeding, but some pathogens are transmitted from a mother vector to her offspring via her eggs or embryos. It is dependent on the type of mouthparts with suitable inoculating devices, example stylet in the mouthparts, is with the vector. Vector stylets as the critical region where the virus is transferred from vector to plant during feeding. A bridging polypeptide molecule links virus particles to the epid's stylet tips. A virus can genetically code for its own linking molecule, that is called helper factor, or provide a helper factor to aid the vector transmission of another virus. However, how the virus detaches from the vector mouthparts is not understood. Several researchers have reported on spreading of vector-borne zoonotic diseases due to human activities. The rapid changes in land use, trade globalization and social upheaval are causing resurgence in zoonotic diseases across the world. Vector-borne diseases are multifactorial in their appearance. Various factors such as host animals, insect and man are influence the incidence of vector-borne diseases. Few important vector-borne zoonotic diseases include dengue fever, Lyme disease, plague, West Nile virus, etc. Latent period. Vectors that increase their transmission rates over time have latent period. The time between a vector's acquisition of the pathogen and its first transmission to plants. Some vectors never transmit a pathogen until days or weeks after they first acquire it. In contrast to this, some vectors need no latent period. Transmission can occur immediately after acquisition. Various vectors are particularly sensitive to meteorological conditions. For example, Anopheles mosquitoes only transmit malaria where temperature routinely exceeds 60°F. Temperature influences the proliferation rate of the mosquito as well as the maturation rate of the parasite within the insect body. Mosquitoes live only a few weeks. If warmer temperature rises, the odds that the parasite will mature in time for the insect to spread the protozoan to humans. Global climate change has already altered the species ranges of number of animals and plants. Further, change may increase the range of the mosquito vectors that transmit disease. This could expose 60% of the world's population to malaria carrying mosquitoes. Unfortunately, 45% human population now resides in a zone of potential malaria transmission. Moreover, weather patterns can also influence the number of vertebrate animals serving as reservoirs for various pathogens. Medical practices, the adaptability of microbes, global travel, crowding, human susceptibility, alternate vertebrate hosts, insect vectors and climate are just some of the factors that influence the emergence of vector-borne diseases. In most cases, the interplay between multiple factors must be understood. War and famine also set up conditions that lead to the emergence of vector-borne diseases. Prevention and control of vector-borne diseases. Prevention and control of vector-borne diseases require a multifaceted approach that involves all the interrelated factors which contribute to the persistence and survival of disease-causing agents. The World Health Organization states that control and prevention of vector-borne diseases are emphasizing integrated vector management, which is an approach that looks at the links between health and environment, optimizing benefits to both. In April 2014, WHO launched a campaign called Small Bite, Big Threat to educate people about vector-borne illnesses. WHO issued reports indicating that vector-borne illnesses affect poor people, especially people living in areas that do not have adequate levels of sanitation, drinking water and good housing. Therefore, individuals from many disciplines such as biologists, chemists, statisticians, atmospheric scientists, environmentalists, ecologists and public must join their hands to work together for preventing and controlling the insect vector-borne diseases in plants and animals. Sanitation is most often used to prevent vector acquisition. Vigilant sanitary measures for quick removal and destruction of diseased plants. It helps eliminate the secondary infections by fungus and to reduce vectors population. Ensuring that new plants are free of the targeted pathogen by using certified propagating materials. Physical isolation of crops from exposure to vectors can be used in greenhouses or by growing crops in regions where the vector populations are very low. Modifying plantations dates to avoid peak periods of vector activity if these can be reliably predicted and do not cause unacceptable losses in yields or quality. Using resistant varieties of plants is a highly effective control if genetic resistance is available. Cultivating new varieties to increase the availability of resistant varieties against diseases. Timely application of insecticide on the crops shortly after they fly into the crop can kill effectively the vectors like aphids. Follow integrated vector management practices on vector and host level. Integrated vector management is achieved by using physical, mechanical, chemical and biological methods to prevent the development of immature stages. Use growth inhibitors or vector growth regulators if possible at different stages, namely egg, larva, pupa and adult, to prevent further development. Further, using biomolecules, example proteins which are expressed by the parasite or pathogen, example in case of malarial parasite, during different stages of their life cycle in the host body is one of the elite methods to be followed during diagnosis of vector-borne diseases. Insect vectors are present in different orders such as Aenuflora, Coleoptera, Dermaptera, Diptera, Hemiptera, Homoptera, Orthoptera, Cypanoptera and Thysanoptera. The insects belong to these orders are known to carry different pathogens such as bacteria, virus, fungi and parasites, example protozoan flagellates and nematodes, and transmit various diseases in plants, animals including man. All these vectors have modified mouthparts which help suck blood or body fluids in animals or cell sap or vascular fluid from vascular tissue in plants. During this course of act, vectors modify mouthparts that include small stylets, allow them to introduce pathogens, example virus, bacteria, fungi or parasites, example protozoan flagellates or nematodes, into host's body, example animals or plants, with minimal feeding damage. Aphids, whiteflies, leafhoppers, plant hoppers, mealybug, mosquitoes, flies, lices, fleas, louses, thrips, beetles, grasshoppers and airwicks transmit different type of dreadful diseases among humans, domestic animals and plants at different parts of the world. Insect vectors infestation can be noticed by observing the infested animal or plant which shows specific symptoms. Vectors bite develop prominent symptoms which are species specific, and their life cycle is also quite interesting to understand their prevalence. Vectors bite could develop secondary infections while transmitting the pathogens into the host body. Therefore, integrated vector management practices should be followed to control the insect vectors population effectively at their diversified habitat around the globe. Thank you.